Those of us who have been in the industry now for 3 decades, we remember the promise of the genomics revolution. Sequence the human genome and all will be revealed. We will understand health, we will understand disease, we will understand therapies. And that promise didn't really take hold. The reason for that is context.
Context in microscopy can be viewed in, let's say, 3 ways. You have the ability to look at both 2D and 3D, so bringing context in the dimensions that we look at. We also have context in temporal aspects, so bringing in the time aspects, so watching biology take place over time. And there's also a context in scales, being able to go from looking at something in subcellular resolution, maybe the distribution of proteins within a cell, being able to move that up into a group of cells, into a tissue, into an organ, and then ultimately into an organism. And being able to look at all of those aspects delivers the context that we need to get to those breakthroughs in understanding.
One product within our portfolio we are really proud of as it relates to this is the CellDive.
CellDive is a spatial multiplexing tool. It's an end-to-end workflow that uses iterative staining and destaining rounds to go past the usual limitation on the number of biomarkers that can be analyzed in a single experiment, even up to 60 biomarkers and more. CellDive and other spatial multiplexing tools are particularly useful in any situation where you require a lot of biomarkers to precisely identify cells or biological processes in tissue. For example, there are immune cell subtypes that can require 5 or even 10 biomarkers to precisely identify. So in an experiment with limited biomarker budget, that can be a problem, and CellDive allows the researcher to go past that. And it can be especially important for not only identifying cell types, but also adding positional information and spatial context. CellDive is especially important in the immuno-oncology field, where mapping the tumor microenvironment, that is, all the various cell types, tissues, and systems that are converging on the tumor, mapping that's particularly important for understanding why some patients respond to certain therapies, why some don't. And maybe also for understanding why some patients experience a certain clinical course of a given cancer type, and some patients experience a much worse course.
What do we do to democratize some of these tools so all researchers can access the images they need to provide that critical context? For that reason, we developed the MICA platform that can allow someone without a lot of expertise to consistently get high quality, reliable, repeatable images with a push of a few buttons. Not only did we kind of create it in wide field for some of the simpler imaging, but also the wide focal, so that the capability of confocal imaging, which has typically required expensive instruments and a lot of imaging expertise to generate good images, is now available to all researchers. The challenge has always been, how much can you see without harming your sample and thereby impacting the image and the data you get? We refer to that as phototoxicity. So this is one of the challenges that we as an imaging company are working to solve. And we're particularly excited about the Tau-STED Extend, which is really gentle, live cell, super resolution microscopy. So researchers can get the images that they need and the data that they need without dealing with this phototoxicity, which would harm their sample and, probably most importantly, impact their data. Within that spatial context now, we know that we need resolution, precision, and reliability so that the image we take today and the information we get today is the same information we would get tomorrow if we took it again.